Self-organization within living systems can emerge through numerous spatiotemporal interactions between entities to achieve a degree of order in the form of pattern formation. Here I would like to take one such example of spatiotemporal pattern formation, the developing zebrafish vertebrate system. The vertebrate embryo segmentation is a process wherein the embryonic body axis is subdivided into repeated segmental structures called somites as depicted in the movie on the left. The presomatic mesoderm or the PSM is the unsegmented tissue until the last formed somite consisting of the cellular oscillators that form the segmentation clock. This segmentation clock is responsible for the sequential and periodic formation of the somites. So the segmentation clock consists of cell autonomous genetic oscillators involving cyclic genes of HER1, HER7 and delta C. For simplicity, we show the HER here. The neighboring cellular oscillators are synchronized via intercellular interactions through cell-to-cell contacts, and this is arising from the delta notch signaling. This interaction causes the cells to remain synchronized by aligning their oscillation phases and thereby setting the somatogenesis rhythm. At the same time, as we can see here in the cartoon, the movement of cells is entirely random in the PSM and they move at a much higher rate in the posterior PSM, as we will see in the next minute. Hence, we asked how this random movement influences the synchronization properties among the PSM cells. To answer this question, we performed confocal live imaging of cell mixing and synchronized oscillations within the PSM. The cell movements are random here and exhibit three-dimensional movements as seen on the left. At the same time, a rapid and dynamic molecular wave involving transcriptional oscillations of cyclic genes synchronously traverses the tissue from posterior to anterior, resulting in the sequential and periodic formation of the somites. Here the line HER1 only marks the presomatic mesoderm cells, hence the somatic boundary cannot be seen. This is not expressed in the somites. The synchronization is, however, achieved via cell-to-cell coupling, whereas the cell movements is random. Hence, we ask how this movement affects the synchronization of these oscillations. So, to answer this, we first went on to analyze just the cell movements, and one 3D reconstruction of a single stack shows that these cells are tightly packed and they are highly juxtaposed nuclei within this tissue. So we developed customized algorithm that automatically identified the nuclei, segmented the nuclei in 3D and quantified cellular features within such a densely packed environment. Then using these 3D cellular features from the segmentation results, we customized a 3D centroid tracking algorithm based on the nearest neighbor links and computed the spatial and temporal cellular features. Here is the 3D tracking result showing the cellular tracks throughout the PSM. Next, we simultaneously imaged cell movement, here shown in gray, and the HER1 expression shown in blue, marking just the PSM cells. We tracked single cell HER1 oscillatory expression from the 3D cellular tracks within the PSM. The Hilbert transform converted the oscillator expression into its corresponding phase. This led us to computationally analyze cell movements and oscillating protein dynamics at single cell resolution throughout the PSM and achieve thereby system level parameters for a quantitative understanding of the in vivo spatiotemporal behavior of oscillators. The coupled frequency profile varied along the posterior-anterior axis, and consequently spatially distinct periods were observed as shown here. The distribution of oscillators' phase differences showed relatively greater width in the tailbud PSM compared to the presumptive anterior somite. We also computed the time evolution of the phase order parameter, a key quantity representing synchronization levels among an ensemble of oscillators. Our results indicated HER1 synchronization levels oscillate in the anterior presumptive somite region. That could be crucial possibly for stopping the HER1 oscillations prior to formation of somite boundaries. Here we can see that the phase order parameter can take a value of 1, which occurs when all of the oscillators have similar on or off timing states, and the cell movements in this region are very low. On the other hand, in the tailbird and posterior PSM cells, the oscillators tend to have higher velocities, and this could actually help them to exit that zone at a faster rate. We summarize our results with the model showing a gradient in the relative velocity of cellular oscillators along the long axis, with the highest being in the posterior and tailbird PSM cells, a frequency profile along the long axis, and a phase synchronization transition that occurs along this axis as well as a result of this, as seen in the previous slide. We conclude from these analysis that these anterior oscillating order parameter could be important for stopping the HER1 oscillations to form the boundaries, and that the higher movement rates in the posterior PSM could actually be useful for propagating synchronized oscillations towards the anterior. Next, we considered this mixing or movement of oscillators could introduce a dynamic topology throughout the PSM, in the presence of which these oscillators must synchronize. Therefore, the coupling strength might be spatially modulated as the PSM undergoes a continuous phase synchronization transition along its long axis. We analytically inferred spatial phase-to-phase correlation function from the known steady-state distribution of oscillators using the Kuromoto model setup. And we considered the case of heterogeneous frequencies as observed in our data. Our results indicated that we can infer the range and strength of interactions from this model. With this, I would like to thank all the people who made this achievable, the institutions and the funding, and thank you for the invitation.